Abscisic acid is a sesquiterpene hormone that does not play a role in plant growth promotion like auxins, gibberellins, and cytokinins. It plays an important role in regulating processes like induction of dormancy in seeds or in coping with abiotic and biotic stress situations. This hormone was seen to bring about stomatal closure in plants that were subjected to water scarcity, thus protecting the plant from wilting. ABA was also known to play a role in accumulation of seed storage proteins and in protecting seeds from desiccation-related damage during the dormancy phase. In this module, we will first study how this hormone is synthesized, transported, and metabolized. Then we will see how abscisic acid signaling takes place to bring about responses like stomatal closure and how this hormone regulates gene expression required for protecting plants against stress. ABA is synthesized by the terpenoid biosynthesis pathway. In plastids, the 5-carbon compound isopentyl diphosphate is used as a building block to form larger terpenoids. Surprisingly, in plants, abscisic acid is synthesized not from the C15 sesquiterpene precursor farnesyl diphosphate, but is synthesized by cleavage of the C40 hydroxylated carotenoid called zeaxanthin. Zeaxanthin is converted to violaxanthin by the enzyme zeaxanthin epoxidase ZEP. Violaxanthin is then cleaved to form the 15-carbon compound xanthoxin by the enzyme epoxycarotenoid dioxygenase or NCED. This is the first committed step in ABA biosynthesis. Xanthoxin is transported out from the plastids into the cytosol, where it is converted to abscisic aldehyde and then abscisic acid. Mutants defective in ABA synthesis have proved to be very useful in identifying many of the steps in ABA biosynthesis. Inactivation of ABA occurs through the formation of hydroxylated products by the action of cytochrome P450 oxygenases, which are further converted to phasic acid and dehydrophasic acid. ABA can also be inactivated by the formation of glucose esters. These esters are transported to the endoplasmic reticulum or vacuoles for storage, and free ABA is released when required by the action of glucosidases located in these organelles. ABA is synthesized in vascular tissues of leaves, from where it is transported by an intercellular pathway to the ABA responsive tissues like guard cells. Active transport of ABA across cell membranes takes place via ATP binding cassette or ABC transporters. These transporters are involved in active transport of abscisic acid across membranes and is accompanied by ATP hydrolysis. An ABA transporter, ABCG25, has been shown to bring about the export of ABA from vascular parenchyma cells where it is synthesized. Another ABC transporter, ATABCG40, is located in the plasma membranes of ABA-responsive guard cells and plays a role in ABA import, leading to stomatal closure. Import of ABA into cells is necessary because the soluble ABA receptors are located in the cytoplasm. One of the most characteristic responses brought about by ABA signaling is stomatal closure and is seen as a water conservation mechanism when plants are subjected to drought stress. ABA receptors are GTP binding proteins which, when bound to ABA, bring about opening of calcium channels. Calcium entry into cells leads to membrane depolarization and activation of anion channels as well as outward rectifying potassium channels. This leads to a loss of turgor in the guard cells and causes stomatal closure. High levels of intracellular calcium also inhibits the plasma membrane H plus ATPase and causes alkalinization of the apoplasm. This also leads to activation of anion channels and potassium efflux channels, causing stomatal closure. ABA also plays an important role during seed development, and ABA levels are seen to increase during two phases, namely during accumulation of reserves and during the desiccation phase. ABA leads to inhibition of cell division and growth that would otherwise lead to precocious germination or vivipary. Several ABA responsive genes like leafy cotyledon or LEC, ABA insensitive 3 or ABI3 play a role in bringing about this inhibition. At the same time, the developing seed accumulates reserves, and the genes coding for the synthesis of seed storage proteins are shown to be positively regulated by abscisic acid. As seed maturation ends, the desiccation phase of seed development starts. ABA plays a role in the induction of dormancy and desiccation tolerance by activating late embryogenesis abundant or LEA proteins that protect the embryo from desiccation-induced damage by preserving cell ultrastructure. How does ABA bring about the effects that we just saw? For doing so, the cell has to perceive and respond to ABA. ABA receptors belong to a family of small proteins called pyrobactin resistance or PYR or regulatory component of ABA receptor RCAR proteins. Binding of ABA to the receptor leads to inhibition of a specific protein phosphatase PP2C, which is a constitutive negative regulator of a family of kinases called sucrose non-fermenting or SNF1 related protein kinases. Autophosphorylation of SNRK2 activates the kinase, which then phosphorylates several downstream transcription factors that include abscisic acid response element binding factors or ABFs. The ABFs belong to the BZIP superfamily of transcription factors. They bind to the ABA response element or ABRE present in the promoters of ABA response genes and leads to their activation, especially under abiotic stress conditions. ABA signaling is also known to occur through modulating the levels of transcription factors ABI3 or ABI5 through ubiquitin-mediated proteasome degradation. For example, ABI3, which is a negative regulator of ABA responses, is targeted for degradation by ubiquitination involving the E3 ligase ABI interacting protein AIP2, whose levels increase in presence of ABA. On the other hand, ABI5, which is a positive regulator of ABA responses, is not allowed to accumulate in cells due to its degradation by another ubiquitin E3 ligase keep on going or KEG. In the presence of ABA, the degradation of ABI5 is inhibited by auto-ubiquitination of KEG, thus allowing ABI5 to bring about activation of ABA response genes. To summarize, the plant hormone ABA affects various plant processes through a complex signaling pathway. Some rapid responses, like stomatal closure, are mediated without altering gene expression. They depend on the increase in ABA levels accompanying stress. On the other hand, slower responses involve regulation of gene expression. ABA is therefore an important hormone that plays a role in both normal developmental processes as well as the defense processes involved in increasing stress tolerance.